Hello everyone, today we are going to talk about blood, extremely important topic for the future doctor. So if you want to be a really good specialist, you should work hard this week. Let's start directly from the red blood cells. Besides, blood contains plasma, its liquid portion, but today we pay attention predominantly to the blood cells, and we start from the most numerous red blood cells or erythrocytes. At the beginning I want to highlight that this week and this lesson you have to prepare exact numbers, and several parameters should be learned by heart. For example, the number of erythrocytes per 1 liter of the blood. So notice that there is a difference between the males and females, and you need to know not just something like average concentration or something like approximately 6 into 10 power 12. You should know the exact range, 3.9 up to 6 into 10 power 12 per 1 liter as for the male, and 3.7 up to 5.5 into 10 power 12 for females. Notice that female organism the concentration of red blood cells is a little bit lower due to the effects of estrogens. So please, you should learn by heart those parameters. You will be asked, and this is the basic knowledge. Even for the minimal mark, you should know this information. Okay, so another two terms should be familiar to you. These are erythrocytosis and erythropenia. Erythropenia, when the amount of erythrocytes is decreased, and erythrocytosis if their number is increased. Also, another important parameter is the diameter of the red blood cell. The average diameter is 7.5 micrometers. 7.5, this is the normal red blood cell. If more than 8 micrometers in diameter, then we call them macrocytes. If less than 7, then we call them microcytes. And we'll discuss it later. Another important information is the shape. Erythrocytes, they have biconcave shape, and it helps them to perform their function perfectly. So if the shape is changed or distorted, such erythrocytes are not very effective and they should be destroyed. So now we have to understand how this shape is maintained and what helps erythrocyte to be so perfect in shape. First, there are 3 groups of proteins, and the first of them is incorporated into the plasma membrane. So these are integral proteins, and they are shown here with a yellow and purple color. So integral proteins, these are glycophorin and band 3 proteins. Please remember them. So 2 transmembrane proteins. Why they are so important for you as a future doctor? Because they could be glycosylated in different way, and the pattern of their glycosylation determines the blood group. You know that there is the AB0 system of blood groups, and different individuals, they bear different antigens, and those antigens are exactly determined by sugar molecules on the surface of glycophorin and band 3 proteins. Okay, so we have 2 transmembrane proteins. After that, if we look inside the red blood cell, we'll find just under the membrane submembrane complex made up of spectrin molecules. Spectrin molecules, shown here alpha and beta, dark and light blue color, they form a scaffold of the red blood cell. So the shape is determined not by the plasma membrane, it is soft. You remember that liquid mosaic model, it implies the liquid nature, more or less soft. But the shape is determined exactly by the spectrin. It forms a network of the biconcave shape. And plasma membrane is fixed to this scaffold with the help of 2 proteins. These are anchorin protein. It links spectrin to the band 3 protein, and another one is the band 4.1. here it is labeled band 4.1. It anchors the glycophorin to the same spectrin molecules. So actually there are many more proteins in this scaffold, but you should know at least those 3. Why they are so important, and in this slide they are given in text, because mutations of every protein results in the distorted shape of the red blood cell and its malfunction. So actually there are many more proteins, but those we put in this slide are extremely important, and you will be asked about them. Okay, let's move on, and now we have to discuss what problems could be with the shape and size of erythrocytes. As I told you, some erythrocytes, they could be less than 7 micrometers in diameter. Some of them could be larger than 8 micrometer. So they could be micro and macrocytes. If there are 25% of such erythrocytes in the blood smear, it is a normal circumstance, it is a normal situation. But if more than 25% of all red blood cells are represented by macro or microcytes, then we call this condition anisocytosis. Anisocytosis means N like D9, iso means equal, and cytos means cells. So if to translate from Latin, it will be not equal cells, not equal in diameter. So you have to know the definition of this condition. And anisocytosis is a condition when more than 25% of red blood cells are represented by micro and macrocytes. You remember the average diameter is 7.5. Another term and another pathological condition is called poikilocytosis. Poikilos means diverse, and this condition is defined when more than 20% of red blood cells are distorted in shape. So if more than 20% are represented by not by concave discs, but of another shape, we call this poikilocytosis. When it occurs, when you know that average lifespan of one red blood cell is nearly 120 days. After that, red blood cells are destroyed either in the spleen or in the liver, maybe in the red bone marrow. And how they are recognized, why they are destroyed. Old erythrocytes, senile erythrocytes, they are usually distorted in shape because they undergo the process of oxygenation along their life, and they acquire some spikes, they become spherical, and so on. So old erythrocytes usually are of abnormal shape and they should be destroyed. And if they are not destroyed, for example, if spleen is removed, if during surgical intervention spleen was removed from the patient, so in such patient we'll find lots of old red blood cells in the blood. Old red blood cells are distorted in shape, and we call this condition poikilocytosis. Notice that up to 20% of erythrocytes could be distorted in shape. This is the normal situation. But if more, then this is the pathological condition. Okay, one more term is connected with the red blood cells. This is reticulocytes. Reticulocytes, these are immature red blood cells, and up to 1% of all circulating red blood cells could be represented by the reticulocytes. Why we call them reticulocyte? Because they have remnants of the endoplasmic reticulum in their cytoplasm. You know that usually all organelles are destroyed, and red blood cell, it has no nucleus, it has no mitochondria, and it contains only hemoglobin inside. But immature red blood cells, they have some endoplasmic reticulum. That's why they are called reticulocytes. So why it is important to know about them? Because if their percentage is increased, this is the sign of hemorrhage and bleeding. So after the bleeding, when organism has lost lots of red blood cells, immature red blood cells are injected into the bloodstream as a compensatory reaction. So if you see in patients blood analysis increased amount of reticulocytes, you suspect bleeding or hemorrhage in the past. Yeah. Next, as for hemoglobin, what's there inside the red blood cell? Hemoglobin, this is the iron-containing molecule, and it is represented by 4 subunits. and it carries oxygen and carbon dioxide under normal circumstances. So the combination of oxygen and hemoglobin is called oxyhemoglobin, and it is of bright red color. When hemoglobin carries carbon dioxide, it turns into dark blue color, almost black, and we call this combination carb hemoglobin or carbaminohemoglobin. That's what you have to know. And one abnormal composition, one abnormal combination could be formed. This is the combination of hemoglobin and carbon monoxide. This is exactly what's happening during the fire, and people, they die by inhaling the carbon monoxide. Why? Because carbon monoxide, it forms some irreversible combination with the hemoglobin. So if oxygen could be reversibly bound to the hemoglobin and carbon dioxide as well, then carbon monoxide, it binds irreversibly and it blocks hemoglobin, and such form of hemoglobin can no longer carry neither oxygen nor carbon dioxide, so it is spoiled. That's why only blood transfusion could be performed to treat this condition. So please do not confuse carboxyhemoglobin with carb hemoglobin, and just you have to understand the difference and why carbon monoxide is so dangerous and poisonous for humans. Okay, let's move on, and one more form of hemoglobin should be learned, should be discussed today. So in adult organism, we have adult hemoglobin, hemoglobin A. Adult hemoglobin, it has 2 alpha and 2 beta chains, but before the birth, during the fetal period, in the fetus, a special form of fetal hemoglobin circulates. And this hemoglobin, it has 2 gamma chains instead of beta chains. And what is especially important, that fetal hemoglobin, it has greater affinity for oxygen than adult hemoglobin. So when they compete for the same oxygen molecule, fetal hemoglobin captures this oxygen more readily, more quickly, more effectively than adult hemoglobin. And it is very important because before the birth, fetus, it gets oxygen from the maternal blood. So it gets oxygen not from the air, not from the outer environment, but from maternal blood. So it has to detach oxygen from the maternal hemoglobin. For this purpose, fetal hemoglobin has greater affinity for oxygen. And it is important because after the birth, fetal hemoglobin is massively broken, and so transient jaundice takes place. So just after the birth, nearly the 3rd day, in newborn children, jaundice could be observed. That's because of the massive breakdown of fetal hemoglobin. You know that bilirubin or bile pigments, these are the products of hemoglobin degradation. So you should be aware about this condition and about this phenomenon. Okay, let's move on. We have completed with erythrocytes. You know that their function, of course, to carry the gases and some other substances, but let's investigate in more details leukocytes. Leukocytes, these are white blood cells, and they are much less numerous, and there is another parameter you should learn by heart. This is the range of their concentration from 4 to 10 into 10 power 9 per 1 liter. What does it mean, 10 power 9? 9 zeros, yeah? And notice that if red blood cells were arranged into 10 power 12, then for leukocytes we have 10 power 9. So red blood cells are 1,000 times more numerous than leukocytes. That's why our blood is red, not white, because red blood cells predominate. For 1,000 of erythrocyte, only 1 leukocyte is present there. Okay, so if there are more leukocytes than 10 into 10 power 9, then we call this condition leukocytosis. If less than 4, then we call this leukopenia. And now let's talk about their classification. This is the bold question, and this is the basic knowledge. So all leukocytes are subdivided into 2 broad groups, agranulocytes and granulocytes. Besides both groups, they have granules. Primary eosinophilic granules, these are the lysosomes. So lysosomes are present in both groups, but they are almost invisible with the light microscopy, only under the high magnification. But granulocytes, except primary granules, they also have specific granules. They are much larger, and they could be observed with the light microscopy, even with a low magnification. So that's why they are called granulocytes, because they have large specific granules. And also another feature of all granulocytes, this is the segmented nucleus. They have their nucleus segmented into several fragments, especially neutrophils. They have the most segmented nucleus. Okay, and now let's consider every leukocyte one by one. And besides, notice that you have to remember the percentage of every form in the blood smear. This is what we call the leukocyte formula, and you have to be aware about those parameters. But before we talk about every leukocyte in details, let's consider the composition of those primary eosinophilic granules, because they are present in all leukocytes. So what do they contain? Primary eosinophilic granules, these are the lysosomes, and they contain several special set of enzymes. First, myeloperoxidase. Myeloperoxidase, this enzyme, it generates the toxic molecules. For example, the hypochlorite and chloramine. These are chemicals destroying bacteria non-specifically, because just these molecules are just chemicals. Okay, then special enzymes like defensins and catalycidin. These are the two enzymes breaking down the bacterial membrane. So the bacterial membrane, is broken down with the help of those two. Then lysozyme. Please do not confuse with lysosome, because many students are confusing. Lysozyme, this is an enzyme. That's why lysozyme, okay? And this enzyme breaks down bacterial cell wall. You know that bacteria accept membrane. They have additionally the cell wall made up of sugars or carbohydrates. And those carbohydrates are broken down with the lysozyme. So lysozyme, they break down cell wall, while defensins and catalycidins, they break down membrane. Together they break down everything. And acid hydrolases, they hydrolyze organic molecules like lipids, sugars, nucleic acids, specifically. Okay, so primary granules, they contain a cocktail of enzymes breaking down bacteria non-specifically. And now let's investigate every leukocyte in details and understand its exact function, because they all are destined to perform quite different function. They all are like an army fighting against invaders, but every department, every subgroup, it has its exact definition and its purpose. So let's start from the most numerous soldiers of our blood. These are neutrophils. And the percentage you should remember exactly, 65 up to 70% of all leukocytes are represented by neutrophils, up to 70%. First, we should discuss its shape and structural features. So they have the nucleus segmented, and also specific granules. Specific granules are small, and they are stained with basic and acidic dyes. That's why neutrophils are called neutral. Neutral because the granules, they are acidic as well as basic. That's why neutral. So small specific granules and highly segmented nucleus. What is especially important that in neutrophils of female organism, a special segment is present which is called bar body, and it is of drumstick appearance, and that's what helps in forensic medicine to detect the gender of the suspect by the blood smear. So if in a blood smear we see this drumstick, so we know that this is the female blood. If there is no, then it should be male blood. And this is how the gender is very easily detected. Okay, now what about their functions and specific granules? Let's first look at the function. Function, they fight against bacteria, simply phagocytosing them. You already know what is the phagocytosis, and it is performed with the help of the actin cytoskeleton. So neutrophils, they engulf bacteria, and phagosome is fused with a specific granules, and in this way bacteria is destroyed inside the neutrophil. And they are the first cells to arrive at the site of infection. So if anywhere in our organism bacteria try to colonize the tissue, so neutrophils are attracted and they come and try to destroy bacteria. How they fight against bacteria? First, they have primary xerophilic granules with a previously discussed set of enzymes. Also, they have specific granules, and let's discuss what they contain. Specific granules, they contain alkaline phosphatase, collagenase, gelatinase, elastase, and some other enzymes. So why they need collagenase and gelatinase elastase? Not to fight against bacteria, but to break down the basal lamina of the blood vessel. So neutrophils, they circulate in the blood for several days, and if they feel the bacteria in the tissue locally in this site, they get attached to the endothelium, and through diapedesis they come out of the blood vessels and they enter the tissue. And to come out of the blood vessel, they should cross the wall, blood vessel wall, and to do that they need to break down the basal lamina with its collagen. You remember previous lecture, and you know that collagen is present in the basal lamina. So specific granules are needed even not to fight against bacteria, but to migrate in the connective tissue, to break down basal lamina, just to break down its own tissue. That's why if neutrophils are hyperactive, they are really dangerous, because they can destroy not only bacteria, but our own tissues around bacteria. Okay, so sometimes we need to pacify them. So tertiary granules are also mentioned, and there are two types, so with the phosphatases and metalloproteinases. Actually, metalloproteinases are the same as collagenase, gelatinase, and elastase. So they have the same function, but enzymes are packed and they are sorted into different granules. Some of them, they contain only metalloproteinases and only phosphatases. Just remember that. Okay. Now we have to discuss what factors stimulate neutrophil immigration to the site of inflammation, because it is very important. How neutrophils are attracted, because if this mechanism doesn't work, neutrophils can't arrive, can't help to deal with invaders. First phase of neutrophil immigration is when neutrophil is circulating in the blood. Inside the postcapillary venules, they feel the presence of selectin molecules. From the previous lecture, you remember that selectin, these are a group of molecules to interact with immunocompetent cells. So when neutrophils pass by the endothelial cells in the blood vessel, and if it see those selectin molecules on the surface, it gets captured and starts to roll along the endothelial surface. Something like small hooks capture it and slow down its movement. When it rolls along the surface, it is affected by special chemokines produced by endothelial cells, and those chemokines increase the expression of integrin molecules on its surface. So the second step is when neutrophils, they get firmly attached to the endothelium with the help of integrins. So nothing difficult, selectins are needed just to slow down the passage of the neutrophil, and further integrins are needed to fix neutrophil to the blood vessels wall. Then the next phase is called diapedesis. Diapedesis is when neutrophil span the blood vessel wall. It migrates out to the tissue where bacteria are present. So to do that, neutrophils should either break down intercellular junctions and to send its extension, its process between the endothelial cells. This is what we call the paracellular pathway. When neutrophil, it immigrates between the cells, when it squeezes between the two neighboring cells of endothelial tissue. Or, as an alternative, some holes or pores could be formed inside endothelial cell, And in this way, neutrophils, they pass through the endothelial cell. That's what we call transcellular pathway. Again, nothing difficult. So neutrophil could migrate either between two endothelial cells or through the endothelial cell, just making holes in it. And it is important in different circumstances to differentiate processes. Okay, so first selectin molecules work, then integrin molecules, and after that diapedesis takes place. Okay, now we have to discuss how neutrophils do recognize their victim, how they distinguish bacteria from the cells of the same organism. To do that, they are equipped with a set of receptors on their surface. First, these are Fc receptors. They bind to the constant region of immunoglobulin G or antibodies. And in this way, neutrophil phagocytose everything what is covered with antibodies. They don't think what substance, the nature of the substance. They just see if this object is covered with antibodies, then they should phagocytose it. And to recognize antibodies, they have Fc receptors. Then they have complement receptors. Complement is another group of proteins, and complement, it binds to the complexes of antigen and antibody. So the neutrophils, they can recognize complement, which recognizes antigen antibody complexes. So in this way, the same process takes place. With the help of Fc receptors, neutrophils, they recognize antibodies. With the help of complement receptors, they recognize complement bound to antibodies. So they recognize antibodies indirectly with the help of the complement receptors. Then scavenger receptors, they bind to special molecules on the surface of gram-positive and negative bacteria cells. So with the help of these receptors, neutrophils recognize bacteria directly, not through antibodies on its surface, but directly bacteria cells. And another very important group of receptors, these are toll-like receptors, and there are different types of them, and these receptors are destined to recognize bacteria and viruses because they can distinguish special molecules, they are called pathogen-associated molecular patterns, present only in bacteria or viral not cells, viral particles. So some molecules are present only in bacteria. They are never found on the surface of mammalian cell or eukaryotic cell. And such molecules characteristic only for prokaryotic organisms or viral particles are called pathogen-associated molecular patterns. Please remember this abbreviation because it is very widely used in immunology and you have to understand it. So toll-like receptors, they recognize those pathogen-associated molecular patterns present only in bacteria and viral particles and stimulate their phagocytosis. So let's summarize how neutrophils do recognize bacteria, either through antibodies or through complement antibodies complexes, through direct recognition of the bacteria cell by scavenger receptors, or with the help of toll-like receptors. Again, they recognize bacteria because it has a special patterns present only on the surface of bacteria cells. Okay, let's move on. And now we are talking about, we discussed how neutrophils arrived, immigrated to the site of infection, how they recognize bacteria, and what does it do with the bacteria, what is the mechanism of its action, how it kills bacteria. And they have a special mechanism, oxygen-dependent killing. And this is when bacteria is treated with chemicals, with a set of inorganic molecules, breaking it down very quickly. There are two enzymes you have to remember in this process. These are NADPH oxidase, nicotine aminoadenine dinucleotide phosphate. This is a very difficult molecule. In biochemistry you will learn much more about it, but now just please remember abbreviation: NADPH oxidase and myeloperoxidase. These are the two enzymes helping neutrophil to generate a set of poisonous molecules. NADPH oxidase, it is incorporated in the membrane of the phagosome. It is membrane bound molecule, a complex of molecules even. And myeloperoxidase, this is the enzyme present in the primary eosinophilic granules. You remember these are the lysosomes. So how it works, very easy. When bacteria, green one, is phagocytosed, here we have the phagosome. Then NADPH oxidase, it takes one electron from the NADPH molecule and transports this electron to the oxygen molecule inside the phagosome. In this way, oxygen turns into free radical because now it has its electron unpaired. And if electron is unpaired, then this molecule is very reactive. Then this molecule, it is, with the help of superoxide dismutase, transformed into hydrogen peroxide. And in this way, bacteria is attacked by hydrogen peroxide, by the oxygen radical, superoxide radical, and it is oxidized. And in this way, it is broken down with the help of inorganic molecules. Then myeloperoxidase comes from the primary granules. So primary eosinophilic granules, they get fused with the phagosome, and myeloperoxidase stimulates, as we have discussed, the synthesis of the hypochloric acid, of the chloramine, and from this hydrogen peroxide it creates hypochlorite anion, and in this way it adds some poisonous molecules to this cocktail. So in this way bacteria is destroyed. Nothing difficult. So NADPH oxidase, it generates a superoxide radical and hydrogen peroxide, while myeloperoxidase additionally forms some chloride containing molecules like hypochlorite and chloramine. In this way bacteria is attacked by inorganic molecules and get dissolved inside the phagosome. What is important, if NADPH oxidase doesn't work, then activity of neutrophils is increased, and this is the form of immunodeficiency, and such patients, they suffer from recurrent infection, they have exacerbations of chronic infections, and that's what you have to take into account. And what is also interesting is that NADPH oxidase could generate oxygen not only inside the phagosome but also in the outer environment. That's why neutrophils sometimes they damage the tissue around bacteria cells. Okay, let's move on. Yeah, besides in general, this mechanism is called oxygen-dependent killing, because you can see that oxygen molecule is used at the beginning to create a poisonous molecules. So, neutrophils left shift. Another term you have to understand. As we have said, neutrophils are the most numerous leukocytes, but sometimes we can find in the blood not only segmented neutrophils, not only mature neutrophils, terminally differentiated, but some immature, which are called young and band. We can see that their nuclei are S-shaped or kidney-shaped, and such forms of neutrophils, they are immature, and they are present in a low amount, up to 5%. But if the percentage of those young neutrophils increases, it evidences the presence of inflammation in the organism, and that's what we call the left shift of the leukocyte formula. Why left shift? Because usually we arrange the stages of cells differentiation from the least differentiated to the most differentiated, and here we can see young, band, and segmented, the three forms, and one form is transformed into another. And if the percentage of these forms increases, that's what we call the left shift. Okay, we'll return to this question next lecture when we have the topic hematopoiesis. And the last fact about neutrophils, even after the death, they continue to perform their function. They fight even when they become dead. What they do, they transform their corpses into the network. They use their chromatin to create a network sodden with those poisonous enzymes. And these networks, they are like a trap. They entrap bacteria cells in the tissue. And this phenomenon is called neutrophil extracellular traps. So after the death, neutrophils, they are transformed into traps or poisonous networks covering bacteria cells and immobilizing them. In this way, they prevent spread of bacteria, their division, and they help other neutrophils to cope with the problem. Okay, besides, even after the death, neutrophils, neutrophil corpses, their remnants, should be phagocytosed by macrophages because they are very, very poisonous. Neutrophils, they contain such poisonous molecules that they should be digested by another cells of immune system after the death. Okay, now let's move on, and now we'll discuss much more simpler information about another types of granulocytes. So, another type of granulocytes, these are eosinophils, and they represent only up to 5% of all circulating leukocytes, so much less numerous. They contain pink granules. That's why they're called eosinophils. Sometimes they're called acidophils. Because inside those granules there is a positively charged material, and positively charged material attracts acids. That's why acidophils or eosinophils. Under the transmission electron microscope, those granules are detected as elongated ellipsoid-shaped granules. And this is the feature helping us to distinguish eosinophils from other granulocytes, the shape of their granules. Okay, nucleus is usually bilobed and could be easily detected. So that's how we recognize eosinophils, by the color of their granules, by the bilobed nucleus, and in the transmission electron micrograph by ellipsoid shape of their granules. Now, let's talk about its function. What do? They fight against multicellular parasites. So if neutrophils, we have just discussed, they were fighting against bacteria by phagocytosis, eosinophils, they fight against multicellular parasites or unicellular but eukaryotic organisms. And if we can see in the blood smear, in the blood formula, that the amount of eosinophils is increased, we call this eosinophilia. This is the sign either of parasitic infection or allergic reaction. What is the role of eosinophils in allergy? We are going to discuss later. So, how they fight against parasites and what they contain in their granules. Specific granules of eosinophils, they contain acid phosphatase, major basic protein, which is rich in the positively charged amino acid arginine. Notice that exactly due to the positive charge of this amino acid, the protein is called major basic, because this amino acid is positively charged, so it is basic. And that's why eosinophilic granules are pink, because they are positively charged, so they attract acids. They like acids, they are acidophilic. Then, also they produce eosin-derived neurotoxin. It destroys, or how it affects the nerve system of parasites, immobilizing them. And also they contain two special enzymes, histaminase and aryl sulfatase, and they together neutralize the effects of basophils. So next slide we'll discuss basophils and find out how eosinophils, they help to pacify them. Okay, so in general, eosinophils should be associated with parasites and allergic reaction. Next, basophils. These are the least numerous leukocytes. Notice that they could be even absent in the blood smear. And they have lots of dense purple granules, so the nucleus couldn't be even detected with a light microscopy because the cell is full with those granules. Granules, they have a special feature which is called metachromacy. Metachromacy means that the color of the dye, when they are stained with a blue dye, they change its color into purple. So metachromacy is the possibility to change the color of the dye. If they are stained with the blue, they turn the color into purple. Okay, so you have to know the definition of the metachromacy and associate metachromacy with basophils. So let's talk about the function and the composition of the granules. Specific granules, they contain histamine, heparan sulfate, and together they help to increase the permeability of blood capillaries, blood vessels, not only capillaries. Why it is extremely important? That's what you have to know to understand a mechanism of action of the broad group of pharmacological drugs. Because exactly histamine is responsible for the edema. And in all inflammatory reactions, in all allergic reactions, we can see edema accompanying them. And this symptom, what we are fighting against. And to fight against this symptom, lots of antihistamine drugs are used. So to understand how they work, you should understand what is histamine, where we can find it, and what is the action, what is the function of the histamine. So histamine, it increases the permeability of blood vessels. It forms something like slits between endothelial cells, allowing blood plasma to leak out of the capillaries. And due to the leakage of plasma from capillaries to the tissues, tissue gets swollen, and this is what we call edema. This is the sign of inflammation or allergic reaction. And this is the function of the histamine. Then heparin. Heparin, it prevents the blood clotting. So just after blood plasma enters the tissue, it doesn't form clots, and it is maintained in the liquid form. Then eosinophilic chemotactic factor. This factor attracts eosinophils. So after basophils realized their effects, after they caused edema, they call for eosinophils. Why? Let's return back. Because eosinophils, they have histaminase, breaking down the histamine. Okay, so there is a logic. Basophils, they produce histamine and call for eosinophils to seize their effect. Because eosinophils, when they come, they break down the histamine. So histamine action should be fixed in the time, and we shouldn't, open blood vessels or increase their permeability for a long period of time. Okay, and then what else is found inside the basophils? Interleukin 4 and 3 and 13, they promote the immunoglobulin E synthesis. That's what is important for basophils to recognize antigens. And leukotrienes, they also constrict blood vessels wall. Besides that, the effects of leukotrienes are neutralized by the aryl sulfatase produced by the eosinophils. So, just to summarize, what is the function of basophils? They do not fight against invaders directly, so they don't fight with foreign agents, but what they do, they increase permeability of blood vessels to call for help. In this way, they attract other leukocytes together with the blood plasma. Other leukocytes, they are more readily immigrate to the site of inflammation. So basophils, they help other leukocytes to immigrate from the bloodstream locally to the region where bacteria are. And after opening blood vessels, after attracting leukocytes with others, they attract eosinophils, because eosinophils, they stop effect of basophils. Okay, so in allergic reaction, what happens? By mistake, basophils, they start to fight against innocuous neutral molecules like the pollen, like the cat's fur, or the dust particles. So these particles are not trying to invade us or colonize. They can't reproduce in our organism. But by mistake, basophils, they start to degranulate and to realize their effects against neutral particles. Then eosinophils come and try to pacify them. That's why allergy is accompanied by eosinophilia. Not because eosinophils are responsible for the allergy, but because eosinophils, they try to stop the effect of allergic reaction. They try to complete it, to seize it. Okay, difficult, I know, but if you want to be a really good specialist, you have to work. Okay. Then, in general, the function of basophils to increase the permeability of blood vessels, and in this way they participate in the inflammatory reactions, and they cause edema. So you should strongly associate basophils with histamine, heparin, and edema. histamine, heparin, edema. Okay, then we go to agranulocytes, another broad group of leukocytes without specific granules. So their nuclei are not segmented and they contain small non-specific eosinophilic granules only. And we start from lymphocytes because they are most numerous among this group. Up to 40%. They represent of all leukocytes. Another diapason you should learn by heart, 20 up to 40%. What about their shape? Lymphocytes, they have a round nucleus with a thin rim of cytoplasm around it. So very small amount of the cytoplasm and very large nucleus occupying almost the whole cytoplasm. This is the lymphocyte. Then we classify lymphocytes according to their specification, and we can distinguish them under the light microscopy, but we distinguish them by the surface molecules. So special methods like immunocytochemistry is needed to detect different set of lymphocytes. Let's investigate them. So the first type is represented by T lymphocytes. T lymphocytes are cells dependent on thymus. So they develop in our thymus, that's why they are called T. And they all bear a T cell receptor which recognizes the antigen. So what we have to keep in our mind is that agranulocytes, this is the part of specific adaptive immunity. We have just discussed granulocytes. They represented innate immunity. From the birth, they fight against just bacteria, helminths, and this is what is inherited. But agranulocytes and lymphocytes particularly, they are the part of specific immunity, adaptive. So they can fight against bacteria only after they first meet it. So just if the organism is in contact with the bacteria, then lymphocytes, they start to work. And they start to fight against it only after the first acquaintance, like that. Okay, so T lymphocytes, what's the difference? Every T lymphocyte, it has its own T cell receptor destined to fight against exact antigen. So the army of T lymphocytes is diverse. And every inflammatory reaction, every military conflict, only a group of T lymphocytes work. Only those T lymphocytes work that can recognize receptors on the surface of invader. Okay, then how we classify T lymphocytes? First, T helpers are considered. T helpers, these are CD4 positive cells, and this is exactly the cell which is affected by the human immunodeficiency virus. You know that these diseases is very severe and dangerous because immunity doesn't work. So with this example of the human immunodeficiency syndrome, you can understand the function and significance of helper cells. Helpers are very important cells, despite they do not fight against invaders directly, but they help other cells to be activated. For example, they help B lymphocytes to produce antibodies. They help eosinophils to arrive to the site of inflammation. So in this way, they manage the inflammatory reaction. They help others. For example, if they come and see the helminth in the tissue, they attract eosinophils. If they find bacteria, they call for neutrophils. If they find viral particles, they call for killer cells. So T helpers, they like managers. They decide what cells to activate, what cells to attract, and just what strategy to choose to fight against this exact invader. Then T killers, or CD8 positive cells. Killers are called so because they kill the same cells, the cells of the same organism. Like in humans, between humans, a killer, it kills another organism, the same creature. Why killers should kill the cells of its own body? So they don't fight against bacteria or viruses, but they fight against the same cells. Because if the cell is virally infected if the cell is malignantly transformed, so it forms a tumor, if a cell is transplanted from another organism, such cells are killed by CD8 killers. And that's the very important function of the anti-tumor immunity. And remember that they are CD8 positive. Next, Gamma delta T cells. These are the intraepithelial T lymphocytes, and they have their T cell receptor represented by gamma delta chains. Usually T cell receptor is represented by alpha beta, but they have gamma delta chains, and they do not recirculate. They stay in the epithelial tissues forever, something like the custom service, yeah. And they, like customs, they wait in the epithelial tissues for invaders, so they are usually present on the border of infection. And what is important to say, that all other lymphocytes, they usually recirculate between different organs looking for their antigen, for the cognate antigen. So they could migrate from one lymph node to another and from different organs to each other, while gamma delta T cells, they do not recirculate. Then the last subtype of T lymphocytes is represented by T regulatory cells. They have a special factor of transcription, FOXP3. That's how they are recognized from others. And their main function to suppress immune reactions. Formerly it was called, they were called suppressor cells. But nowadays the term T regulatory cells is widely used and please notice that and fix in your mind, because in some MCQs you can find maybe T suppressor, but this is an old term. Nowadays it is replaced with the T regulatory cells. So sometimes immune reactions should be pacified. For example, if we have already competed bacteria, if infection is, if infection is over, then all immune cells should decrease their activity, and for this purpose T regulatory cells are needed to pacify immune reaction. Okay, then another group of lymphocytes, these are the B lymphocytes. They have quite another function, as they produce antibodies. Antibodies are needed to specifically interact with the bacteria or viral cells or any other invader. And B lymphocytes produce antibodies only if granulocytes can't cope. So usually neutrophils, eosinophils work, and basophils as a first line of defense. They are non-specific immunity and they try to kill just bacteria because it is bacteria. But if they are not effective, if bacteria use some strategies to avoid this attack, then lymphocytes start to work, because they are more time consuming, energy consuming, but they work more specifically. They start to investigate this bacteria and form antibodies individual to this bacteria cell, and this form of immunity is more effective, of course. Then natural killer cells, as the T killers, they work in the same way. They kill virally infected or tumor cells. And how they perform their function: they produce perforins. Like a bullet, it perforates the membrane of the victim, of the target cell, and through those perforations they inject granzymes. Enzymes stimulate an apoptosis in the target. Sorry. So natural killers actually they stimulate apoptosis in the target. And what's the difference between a natural killer and CD8 killers? T killers, they are activated by macrophages, while natural killers, they don't need to be activated. They just work against special molecules, and they know that those molecules appear on the surface of infected cells or something like abnormal cells. So natural killers are more like a part of innate immunity, while T killers, CD8 positive cells, they are a part of adaptive immunity. Okay, very difficult, but very important for the doctor. And the last information about leukocytes. These are the monocytes, and they could represent up to 11% of all the circulating leukocytes. First, let's look at their shape, how we recognize them. They have bean-shaped nucleus or horse-straw shaped, and these are the largest leukocytes in the blood. They do not contain specific granules, but under the very high magnification we can find azurophilic lysosomes there. So what is the function? Monocytes, when they circulate in the blood, they do not do anything, but once they arrive to the tissue, they are transformed into macrophages. And almost in all organs of our body, there is a special population of macrophages. Like in our lungs, we have alveolar macrophages, osteoclasts in bone, and so on. Sorry. What is the function of macrophages? There are two of them, and the first is phagocytosis. So they resemble neutrophils, they also phagocytose their victims. But what's the difference between the neutrophils and macrophages is that after phagocytosing bacteria, they break them down into antigens, and those antigens they expose on their surface and present them to their lymphocytes. So antigens, just after phagocytosing bacteria, its antigens, bacteria antigens, are used to activate respective lymphocytes. You remember that CD4, CD8 lymphocytes, they are specific, they have T-cell receptor destined to exact antigen. So to activate them, those antigens should be present on the surface of macrophages. Because lymphocytes themselves, they do not decide whom to fight against. To activate them, macrophages are needed. So macrophages or monocytes, they are a very important link between the innate and adaptive immunity. So the function of macrophages is not only to digest bacteria, to fight against bacteria, but to activate lymphocytes, to teach lymphocytes whom to fight against. Okay, because lymphocytes, they are specific immunity, and they should be activated by macrophages. Okay, let's move on. And now we have to discuss the last cellular population of our blood. These are platelets. We're approaching to the end, so do not relax. Then platelets, these are the fragments of another cells, which are called megakaryocytes. We are going to discuss them next lecture. So megakaryocytes, these are the giants of our bone marrow, extremely large cells with a highly polyploid nucleus, and when they send their processes to the bloodstream, tips of those processes are pinched off and circulate in the blood in form of platelets. So platelets are truly, they are not cells, and these are just fragments, tips of megakaryocytes protrusions. The number you should remember exactly, 180 up to 320 into 10 power 9 per 1 liter. That's what you will be asked even for the minimal mark. And if there are more thrombocytes, we call this phenomenon thrombocytosis. If less, then thrombocytopenia, because platelets, another name is thrombocytes. Why both conditions are very dangerous? Because blood clotting is a very important process. If blood clots are formed readily and very quickly, it is dangerous because such clots might block vessels in our brain or heart, causing the brain stroke or myocardial infarction. So it's very dangerous and it causes deaths frequently. If blood clots are not formed just after the even minimal traumatic injury, bleeding doesn't stop. So it results in a blood loss and a massive hemorrhages, and this is another set of problems. So to balance the blood clotting process is very important nowadays, and this is the very relevant purpose of the modern medicine. So there are lots of pharmaceutical drugs destined to regulate and to manage the process of blood clotting. And to understand their mechanism of action, you should be aware about the structure of platelets. So let's investigate it. If we make a cross section through the platelet, we could subdivide it into zones. 4 zones are there, the outermost being peripheral, and it is represented by the cell membrane and its glycocalyx. The surface glycoproteins are very important as they participate in the aggregation of platelets if the blood vessel is broken. Then structural zone, it contains, just under the membrane, it contains a bundle of microtubules, actin and myosin molecules. And together they help to maintain the shape of the platelet. Nothing difficult. Then organelle zone. This is the interior of the cell, and it contains mitochondria, rough endoplasmic, and granules. There are 3 types of granules, and that's what we are going to discuss next slide. Then membrane zone. Membrane, it forms two system of tubules. One of them is called the open canalicular system. Open because it is in communication with the outer environment. So here, invagination of the plasma membrane is called open canalicular system. And these are the fragments of the demarcation channels. As I told you, megakaryocytes, they form platelets by just simply fragmentation. And the process of pinching off is a result of the membrane invagination, and remnants of those membrane invaginations are called open canalicular system. It helps to increase the surface area of the platelet, and it helps to discharge the content of its granules during the process of blood clotting. Then dense tubular system. This is the remnant of the smooth endoplasmic reticulum with a calcium storage. Calcium is very important for clotting process, so platelets, they has a depot, and they sequester calcium inside the dense tubular system. So dense tubular because it contains calcium, it is electron dense. Okay, and the last slide, you will be very glad. We are about to finish. So types of granules and their function. There are 3 types of granules. First, very easy to remember, lambda granules, these are the lysosomes. So just with their digestive enzymes, you should not remember their content. Lambda granules, lysosomes. Then what about the delta and alpha granules? Delta granules, they contain ATP, ADP. You know those molecules, they are usually produced in mitochondria. And serotonin together with the histamine. And in parentheses I put the function. So serotonin and histamine, they're needed for the vasoconstriction, for the constriction of blood vessels. Because just after the damage, if blood vessel is broken, it tries to constrict, stopping the blood flow in this way. So vasoconstriction is a mechanism helping mechanically to block the passage of the blood, to reduce the bleeding. Okay, so serotonin and histamine inside delta granules, they help to stop bleeding by constricting the blood vessels wall. The alpha granules, they contain factors needed for blood clotting, like fibrinogen absorbed from the blood plasma, like a platelet-derived growth factor, like plasminogen, and many others. So alpha granules are needed for the initial phase of the blood clotting. And now let's discuss how blood clot is formed. There are only 4 phases. This is the most last, I promise you. So stages of blood clot formation. First, primary hemostatic plaque is formed by just simply aggregation of platelets. So when platelets, they see the defect in the blood vessels wall, if they see connective tissue through the blood vessels wall, they start to aggregate with each other, and they form a primary hemostatic plaque simply by interacting with each other. Then secondary hemostatic plaque is formed when platelets, they discharge their granules and they cause their fibrinogen coagulation and fibrin formation. Fibrin forms a network shown in these scanning electron micrographs. You see those threads forming a meshwork. This is exactly the fibrin after its coagulation. And fibrin, creating this mesh, entraps the blood cells like red blood cells, leukocytes, and by entrapping them it forms a secondary blood clot or the secondary hemostatic plaque. So if primary hemostatic plaque is formed just simply by platelets, then secondary is formed by fibrinogen foreign blood elements and platelets as well. So it blocks the passage of blood through the vessels, through the damaged vessel. After that, with the time, when blood vessel wall is repaired, when it is regenerated, blood flow is how to say, blood flow is returned. So the blood, this clot, it retracts. So the third stage is retraction. You remember that under the membrane of platelet there is an actin and myosin complex. So by contraction of actin and myosin, blood clot retracts and decreases in its size. And the most last stage, the most last for this lecture, this is the resorption process. So when blood clot get resorbed with the help of lambda granules or lysosomes, so it get removed and the blood clot is removed, and the blood flow again just effectively could be seen in the vessel. So these are the 4 stages, and in more details you'll investigate the process in physiology department, of course, but now you should know at least the basis of this process, the structure of platelets, and the basic stages of blood clot formation. So thank you for the attention. Thank you for watching till the end. You are my hero if you really survived till the end of the lecture, and subscribe to our channel if you do really like our lectures. And thank you one more time. Goodbye.